Welcome to this podcast from Neurogastroenterology and Motility. It publishes original research and topical reviews on basic and clinical aspects of gastrointestinal sensation and motility, as well as brain-gut interactions. It is the official journal of the American Neurogastroenterology and Motility Society and the European Society of Neurogastroenterology and Motility.
Welcome everyone to this month's podcast from Neurogastroenterology and Motility. My name is Adam Farmer and I'm a gastroenterologist at the Wingate Institute in London. And this month it's my great pleasure to welcome Professor Stephen Vanna. Stephen is a professor and director of the Gastrointestinal Diseases Research Unit at Queen's University in Kingston, Ontario in Canada. So Stephen, welcome to the podcast and congratulations to you and your co-authors on your paper entitled Concurrent Psychological Stress and Infectious Colitis is Key to Sustained Enhanced Peripheral Sensory Signalling, which is published in this month's journal. So Stephen, could you give me some background to the epiphenomenon that we know as post-infectious irritable bowel syndrome?
Well, thank you very much, Adam. We're really excited to tell you about our study. Post-infectious irritable bowel syndrome is a very common disorder. I think, as everyone knows, irritable bowel syndrome itself is extremely common, and we believe that about 10% of patients with irritable bowel syndrome start with an enteric infection. And we can usually detect that by history because rather than a sort of insidious onset of their symptoms, it's very abrupt, typical of an enteric infection. And then they clear the infection, but these very distressing symptoms of abdominal discomfort and typically diarrhea persist often for many years. And there's been a couple of notable outbreaks around the world, certainly in the UK and one in Canada, as an example, and we've learned a couple of important lessons from looking at this cohort of patients. One is that is around the predictors of who out of this group might have these persisting symptoms. And one is the severity of the infection and the duration. The other intriguing aspect of this is that psychological factors seem to be an important predictor of whether you may get sustained symptoms. And that's what's led us to our interest in this area.
So you suggest that stress and psychological symptoms may play a role in the development of post-infectious IBS. What's already known around this topic in that context?
Well, we've known for a long time that stress is related to the irritable bowel syndrome and psychological factors. It's, I think, pretty clear now that stress is not the cause of irritable bowel syndrome as it was once thought, but it can be an important trigger, and it can certainly aggravate and worsen symptoms. Now, as you might imagine, stress can affect us centrally through our cognition and amplify our sensory signaling in the brain. But we've been particularly interested in another area, is how the stress signaling through hormones in the sympathetic nervous system might be altering pain regulation that's generating from the gut. And there have been a couple of important studies in the past that have shown, both in animal models and in humans, that indeed stress can have important effects on altering pain signaling. We don't know all the mechanisms behind this, but there's evidence that at least in some patients that it may be related to changes in permeability in the epithelium, which maybe allow more microbiome signaling, for example, to the immune system in the lamina propria. And indeed, even though when we take biopsies from patients and send them to a pathologist and they come back reported as normal, if we start looking for more subtle signs of mediators, such as cytokines or proteases or histamine, these are elevated, and we know from studies that they can sensitize the nerves in the gut. And so we and others have shown in the past that when you add stress to infection, that this can lead to sustained pain signaling in the intestine.
Okay, so how does that bring you to the hypothesis of the study that you performed?
So, as I mentioned, we've known about this potential interaction, but we really don't know the specific details behind this. And this is going to be crucial to understanding what possible treatments could emerge from this. So we were interested to know how intimately stress needs to be related to the infection itself in order to lead to these sustained symptoms. And so we had the hypothesis that the stress had to be occurring at the time of the infection to enable the maximal interaction between the inflammatory mediators and the immune mediators that were being driven by these two pathways, one being the stress hormone signaling and the other being the infection.
So what methods did you use to test this hypothesis regarding the temporal relation of stress to the development of longevity of symptoms?
Well, we've used the Citrobacter odentium model of colitis in mice. And we chose this model because it turns out that Citrobacter rodentium parallels in many ways the effects of E. coli infection in humans, which we know is one of the causes of post-infectious IBS in patients. So it turns out that this organism, this bacteria, has the same attaching and effacing properties as E. coli infection. It causes a self-limiting colitis that lasts for 10 to 15 days, and then it resolves spontaneously. So we were then able to, in parallel with inducing this infection, introduce a stressor at various time points. And the stressor that we've used is called water avoidance stress. And this is a fairly simple model where you place the mouse on a platform surrounded by water. And you do this for an hour a day, and we typically would do it for 9 days in a row. Now, we've shown in the past that when you do that, it does induce an anxiety state in these animals. And we've measured epinephrine and corticosterone and shown that these are elevated. And we've also measured behavioral changes and shown that the animals have evidence of anxiety. So we then combine these two models, and I'll come back to the time points, which are important in a moment. But to measure whether it's altering sensory signaling, we then make recordings from the dorsal root ganglion neurons, the nociceptive neurons that are innervating the colon, and we measure changes in excitability of the neurons. And in particular, for those who are familiar with electrophysiology, we look at changes in the rheobase and the number of action potentials that are discharged to 2 times the rheobase. The rheobase, of course, is the minimal amount of current required to induce an action potential.
So what were your key results?
Well, the key findings were that we first wanted to ask the question, well, if we induce this infection and then we apply a stress at a later time point, does that make any difference than if we just apply a stress alone? And what we found was that there was no effect. In other words, there did seem to be something to this relationship of the timing between the infection and the stressful event. So we then set up our important next experiment, which was the key experiment, where we had 2 groups of animals. In one animal group, we applied multiple stressors around the time of the infection. So this was just before the infection, during the acute infection, and during the resolution stage. And we compared that to the other group that received multiple stressors alone but not infection. And then we looked at the distant time point, which was 40 days, which was well beyond resolution of the infection. And what we found was that in the group that had received the multiple stressors during the time and around the time of infection, they had the sustained pain signaling compared to the group that just received the stress alone. So it fit with our hypothesis that there is an important interaction between the infection and the stress signaling. Now, you might have wondered that, well, maybe you've just induced more inflammation and these animals have sustained inflammation. But we showed that, in fact, that's not the case when you look at their body weight, or when we sent the tissues off to our GI pathologist. He couldn't find, using routine H&E staining, any signs of exaggerated inflammation in the animals that received both the infection and the stress. So we believe that there is an important interaction that's going on between the immune modulation related to stress and the infectious agents.
So that brings me on nicely to what do you think the clinical implications are for our patients, and particularly those who develop infectious gastroenteritis?
Well, I think there's a couple of key messages here. Firstly, one of the things, as I'm sure you're aware as a gastroenterologist, that's challenging for patients and for physicians is understand why they have ongoing symptoms despite the fact that when we do all our testing, everything appears to be normal. And I think this provides a nice explanation for how patients can have sustained symptoms due to this initial insult. And it also might start to help us understand some possible interventions. To give you an example, if we have a patient who has known chronic anxiety, for example, and has an acute gastroenteritis, maybe some more aggressive intervention might prevent them from having chronic symptoms. For example, behavioral therapy or short-term medication. Alternatively, if we know, as this study would suggest, that this and as the human studies have suggested, are a particularly vulnerable group, then more aggressive therapy as well directed towards inflammatory mediators may also help to mitigate from them having the long-term symptoms.
And that will inevitably improve outcomes, one would have thought.
Mm-hmm.
What do you think are the key research questions and knowledge gaps that remain in this field now?
Well, I think that we need to delve more deeply into this relationship, even beyond what we've done. For example, we provided stress to maximize the signal before and during the infection, and understanding better, even in more detail, when the key timing is, might help us to understand the mechanism. And for example, what are the key mediators during this interaction? Are they stress hormones that are signaling to the tissue and amplifying somehow certain proteases or histamine, or are they signaling directly to the neurons? The other speculative aspect of this is that it may lead to exaggerated microbiome signaling, and as you know, this is a very contentious and interesting area. And so one possibility could be that with stress, there may be even greater permeability, which would amplify microbiome signaling. Microbiome signaling itself has been shown to have CNS effects in terms of psychological factors. So this could set up a bidirectional signaling pathway that could lead to ongoing symptoms. So delving into the mechanism and finding out in more depth what the key signaling pathways are, I think will be very important.
And certainly will no doubt provide new treatment targets for the future.
Absolutely, that's certainly the hope.
So with that, Stephen, I'd like to thank you so much for joining us on the podcast this month, and also to our listeners for tuning in. I look forward to welcoming you all again on another instalment of the podcast next month.
Further information about this paper can be found on the journal website. We hope that you have enjoyed this podcast, and we look forward to welcoming you to next